Electrophoresis. Today let us learn about electrophoresis. Electrophoresis is a laboratory technique that we all are familiar with. It is used to separate and analyze biomolecules such as DNA and RNA based on their size. In this technique, smaller DNA fragments move faster while larger fragments move more slowly. When an electric field is applied, nucleic acid migrate through a gel matrix towards the positive electrode because they are negatively charged. As we know, DNA carries a negative charge due to its phosphate backbone. Before performing electrophoresis, DNA or RNA is isolated and precipitated using ethanol. Ethanol helps to remove impurities and concentrates in the nucleic acids, making it suitable for further analysis. Nucleic acids can be separated using different types of gels depending on the size of the fragments. Agarose gel electrophoresis is commonly used to separate DNA fragments ranging from 100 base pairs to about 20 kilobase pairs. For very small DNA fragments, polyacrylamide gel electrophoresis is preferred as it is effective for fragments less than 100 base pair. Polyacrylamide gel have lower porosity compared to agarose gels. Due to this reduced pore size, they can separate very small DNA fragments with high resolution, which cannot be efficiently resolved using agarose gels. Before loading DNA into the gel wells, the sample is mixed with a gel loading dye. This dye helps in the visualization of DNA samples during loading and ensures that the sample properly enters and settles inside the well. Without the loading dye, we cannot confirm whether the sample has entered the well since DNA is colorless. The loading dye contains a high molecular weight compound such as glycerol, which increases the density of the sample so that it sinks into the well. During electrophoresis, larger DNA fragments migrate more slowly while smaller fragments migrate faster and travel farther through the gel. Thus, DNA separation occurs primarily based on fragment size. After electrophoresis, DNA bands are visualized using ethidium bromide staining. Ethidium bromide is an intercalating agent that inserts between the DNA base pairs and fluoresces orange when exposed to UV light. The DNA bands can be observed using a UV trans illuminator. In some cases, autoradiography may be used for detection when radioactive labeling is involved. A DNA ladder is run alongside the sample during electrophoresis. The sizes of the fragments in the ladder are already known. If an unknown DNA fragment aligns with a particular band in the ladder, we can determine its approximate size. Pulse field gel electrophoresis. Pulse field gel electrophoresis is a technique used to separate very large DNA molecules up to 10 megabase pairs in size. Using this technique, we can separate large DNA fragments including entire genomes and chromosomes. In conventional agarose gel electrophoresis, DNA fragments larger than 20 kilobase pair tend to migrate at the same rate and appear as a single diffuse band in the gel. As a result, fragments larger than this size cannot be efficiently separated using normal agarose gel electrophoresis. This limitation is overcome in pulse field gel electrophoresis by applying an electric field that periodically changes its direction with respect to the gel. As the direction of the electric field changes, DNA molecules are forced to reorient their direction of migration. Larger DNA fragments take more time to realign, whereas smaller fragments realign more quickly. This differences in reorientation time allows effective separation of large DNA molecules. In pulse field gel electrophoresis, DNA is embedded in agarose plugs before lysis to prevent shearing of large DNA fragments. DNA is then digested using rare cutting restriction enzymes such as SAL1, which produce small number of large fragments. The electrophoresis is carried out using alternating electric field, typically switched every 90 seconds at 120-degree angles. Separation of DNA fragment is based on both their size and their speed of orientation in the electric field. Pulse field gel electrophoresis is widely used for genotyping, genome size estimation, chromosome number determination, bacterial typing, genomic library construction, and epidemiological studies. It is highly reproducible, discriminatory, and effective technique. However, pulse field gel electrophoresis is time consuming, often taking up to 3 days and requires specialized and expensive equipments.